We did kind of end on the hemoglobin, myoglobin topic, and there were some questions that had popped up. So one that I realized that I didn't talk about at all was this BPG molecule, the bisphosphoglycerate, which for some reason I wanted to call phenyl, probably because I'm an organic chemist. But we're looking at bisphosphoglycerate, which weakens oxygen binding. Okay, so if that's in the hemoglobin, it actually prevents oxygen from binding, okay, or minimizes it, which is a good thing when it comes to hemoglobin. Because again, the whole point of hemoglobin is to transport oxygen. You don't want a super strong affinity with oxygen, because if it's a super strong affinity, no oxygen leaves the molecule. So BPG can help release the oxygen or help allow for a better release of that oxygen from your hemoglobin molecule. So if you got BPG bound, your oxygen affinity is a little bit lower. Okay. Yes?
Just out of curiosity, so there was a particular homework question, and I'm not sure if maybe we could talk about that just briefly. That relationship, when the human body is at high altitude. I mean, there was a specific homework question on that. So.
But the high altitude was BPG?
It asks about where the BPG line would move if you were in high altitude. So, and it almost seems like an inverse relationship. Because you would think it would go, the BPG would go, I don't know, when you say the affinity for oxygen.
So what ends up happening is our 50% saturation, come on, there we go. This is our sat level. There's our 50%. Okay. If we look at the pressure for our oxygen, let's say, I don't even know what that number is, but let's make up numbers, say 30. Okay. If we look at BPG, what ends up happening is our curve comes steeper. So when we look at that 50% saturation, we're at a lower oxygen percentage. Okay. So if we're going to look at shifting between standard hemoglobin and BPG, standard up to your without BPG. So this is your no BPG, and this is with. So you'd be looking at that shift. And that's what they're referencing is that oxygen saturation level. Okay. At lower oxygen saturations, no BPG, you bind oxygen at a higher rate or a faster rate.
Okay.
We got this neat little reaction equation that was given in your textbook, which is very helpful for answering a lot of these questions. Okay, so be aware of using that, because that's going to help you out. Because if we change hydrogen ion concentration, we can now use this equation to say, okay, what happens? So if the hydrogen ion concentration increases, which way does our equilibrium shift? This gets bigger, we shift towards the towards our products, and we'd have less oxygen bound. Okay. So if you remember your equilibrium topics, some of you may not, we can treat equilibrium just as a real quick equilibrium, or K. One option is looking at it as kind of a seesaw. Everything wants to balance out so that we get equal weights on both sides, okay, or relatively equal amount, so we get a flat line, a level equilibrium. If we go through and now increase H+, what are we doing on our reactant side? We're putting more weight on this side. Now it goes out of level, so how do we get it back to level? What we're going to have to do is take some of what we put in and put it on the product side, okay, to re-level it. One of the philosophies that then comes out of this, or students' natural response to this, is then we have to have equal concentrations on both sides. Not true, and all of that is based on wherever I put the fulcrum. Okay, because if I put the fulcrum there, that's not going to be equal concentrations on both sides. Okay, so what equilibrium is really referring to is kind of the location of the fulcrum. Okay, is it situated way on the left side? Okay, for our seesaw, in which case we're going to favor our reactants, okay, because we need a lot more reactants, a lot more weight on the reactant side that keep the product side up, okay, to keep that level. If we shift it way over towards our product side, then what we're saying is we need a lot more products. Okay, so our equilibrium constant will kind of change its value based on that. Okay? So when we're talking about looking at a reaction equation, that's pretty much what we're saying. If we're now at equilibrium, it doesn't matter where that K is or our fulcrum is. Once we're at equilibrium and level, what happens if I increase this or decrease this? How does that change the balance? Okay? That's really what's happening kind of as a bottom line with our equilibrium. There was also a question about what happens with acid concentrations and oxygen uptake or CO2 uptake. And very valid question, because if we go through and take a look at our hemoglobin reaction, what happens when we increase the acidity? So if this value gets bigger, what do we do? Okay, we shift to the right, and we release oxygen. So where might we expect to see a high concentration of H+? In the cells, okay, in our muscles, away from the lungs. Okay, and that's kind of what that equation is giving to us. If we now look to what we were talking about when we first brought up CO2, and what Diana was asking about earlier, was the blood buffering system. And we looked at just the buffering system that we find in blood. Okay. What happens if we increase H+? pH decreases, fair enough. What is that going to do to our reaction? What side of our reaction is going to be favored now? What are we going to shift to?
The left.
The left, which is going to be an increase in CO2. Okay, where would we expect to see an increase in CO2, okay, as a gas at high pH, or sorry, low pH, so high H+? We would expect to see this in the lungs. Okay, kind of interesting because those are opposite relationships. Okay. So depending on what we're looking at and how we're shifting things around, we may end up seeing different systems, right, depending on what we're evaluating. When we first looked at the blood buffering system, all we were looking at is the existence of our buffer. We had no understanding or no interpretation of what was going on with H+ concentration and the transport of oxygen. Okay. What we're looking at is solely the buffer. And in the buffer circumstance, if we increase H+, we increase CO2. So we have to look at that equation. When we move to the actual human body, we're having both of these systems happening simultaneously. Okay? And the whole point of the blood buffering is to maintain the level of H+, so that we don't see a large change. Okay? And subtle changes will affect now how hemoglobin reacts with oxygen. Okay? So while it may seem counterintuitive, we have to realize that it's not just this in one circumstance. We evaluated one equation and said under this one equation, this is what we might expect based on our understanding of where CO2 leaves and where we want O2 to be. Okay. So with each equation, we're going to have slightly different interpretations. Okay. Which one was most relevant to O2 transport? Top one. So if we're really going to be concerned about our oxygen transportation and ultimately our CO2 transportation through hemoglobin, we need to be looking at hemoglobin, and the buffer system is there to just maintain our H+ within sort of levels, and then we don't worry about the CO2. Okay? We won't worry about that aspect because it's being taken care of, first off, by our hemoglobin. Okay? So we have to shift our focus depending on what information we have available to us. Yeah, Jay?
When the CO2 is high and when the bicarb go high, because I work as a respiratory therapist and we use blood gases all the time. And for a COPD patient, when they're considered a 50-50 club, so when they're
50-50 club.
Well, when their CO2 is high, their pH, their blood gases are normal because that's their chronic. So when their CO2 is high, like in 50s, their bicarb is also high, like at 26.
And that's because our buffering system is trying to account for that CO2 increase. So it is immediately trying to shuttle it over into the bicarb to reduce the CO2 concentration. So if your CO2 is going to be high, you're going to get that high as well.
Okay, but because on the, when you say that goes up and that will go down.
It goes, so you have to be careful on how much it goes down by. It's going to go down enough so that it achieves equilibrium. But as soon as I add more of the CO2, I'm going to shift, but do I shift everything in that fulcrum situation?
Just as long as it stays in the normal value, is that what you're saying?
As long as the equilibrium is obtained. So if I have my equilibrium, there's my box, there's that other box, and I add that second box. CO2 concentration has gone up. I'm now out of equilibrium. How do I establish equilibrium? I shift over to the right. What do I shift? The whole blue box?
No,
No.
the thing is,
I'm only going to, no, we can't change what it sits on, okay, because that's dependent on what the reaction is. Okay, that is the equilibrium constant for that reaction. What we end up changing is the concentrations of the species. Okay, so if I put in another blue box here, to reobtain equilibrium, I'm going to have to cut a small section out, we call that in purple, I guess, and shuttle that over to the other side. That means my product concentration goes up. But what happened to the CO2 concentration? Well, if this blue box was CO2,
It goes
goes
down.
down.
does it go down? If the blue box is CO2, okay, it goes down relative to the total amount that I've added, but in comparison to where I started, it's still elevated.
Yeah.
Okay? It's not a complete shift of everything across. So if you have elevated CO2, how much it goes down is going to be relative to what you're comparing it to. To your initial equilibrium, it's going to go up. It's going to have to go up.
But you're gonna see an increase in product.
Yes, you'll see an increase in both because that's trying to reobtain that equilibrium.
Correct me if I'm wrong too, your mechanism also shifts in COPD patients from a CO2 drive into an O2 drive. That's why a lot of those patients you don't put oxygen on because
Yeah, not
eventually
that the rest of it.
that's the reason why.
I got nothing to add to that.
Aren't these just two different systems of equilibrium? Aren't enzymes facilitating this? So you shouldn't really, what happens in one of those reactions, you shouldn't really interpret that as, well, it's going to affect.
Is that going to affect the other reaction? If CO2 concentration changes according to the buffer system, is that going to affect this reaction? Does CO2 change? Is CO2 involved in this reaction?
It does, but I look at it as if it's facilitated by enzymes.
It doesn't matter if it's facilitated by enzymes at all. Okay, the enzymes will affect your equilibrium constant and kind of the balance and how fast it occurs, but you're still looking at that reaction. Okay, that reaction is occurring. If you change the CO2 concentration will change your blood buffering system. It will also change how hemoglobin functions.
So those two reactions will actually kind of occur simultaneously with each other?
Yep.
Okay.
And so what's going to determine which one occurs in preference is the one that happens faster.
Okay.
If one of those happens faster, that's what's going to happen first. Then the other one with whatever's left over, and then do an adjustment. Which is kind of nice because that leads into our next topic, sort of. Okay, other questions on that? Okay. There was a quick question about denaturation versus degradation. Okay. Denaturation can be easily reversible. That does not mean it is, but it can be easily reversible. That's where we're breaking all of the interactions that form our tertiary structure for our protein. Okay, so hydrogen bonds, and actually even secondary, it drops all the way down to secondary. So we break all of our hydrogen bonding interactions. This isn't one of your slides officially. It's just a copy over. You break your hydrogen bonds, your metal interactions, okay, all of that. Okay, even your disulfide bonds, that's happening in denaturation. So you've completely denatured the protein, we're down to primary structure. Degradation, we don't actually have a figure for here. What degradation is that I go in and I see the protein and I say, this piece, I don't want anymore. And I cut that piece off. That is now degradation. Because now if I looked at what happened as my result, I would have both the big long denatured piece and I'd have another piece. Okay? I've split that apart. That's a degradation. I've destroyed, physically destroyed the protein. Okay, so we broke the backbone of the peptide. Okay, not interactions within the backbone, but we actually broke the backbone of the peptide. Okay, so you have two pieces now. We run into this question of reversible and irreversible, and depending on which definition you really want to apply, you can run into slight variations off of this. Denaturation can be easily reversible, because all you're looking at is either breaking or reforming relatively weak intermolecular forces. Okay, so that's not that difficult to reverse. And in the worst case, you've got the disulfide bonds. You can go through the oxidation reduction reaction with some facility to be able to do that relatively easily. Okay. Does that mean denaturation is always a reversible process? No, because there are some proteins that are so complex that they need other proteins to help them fold. Okay. Well, if we go through and denature our protein but then don't supply those other facilitators, our protein can't get back to its original shape. Okay. So the denaturation process can be a bit wishy-washy with our means, if that makes any sense. The degradation process, once you've done it, that is not easily reversible. Is it reversible? You broke a bond. Can we make bonds? Yeah, we can make bonds. Is it easy? No. Okay, not by any stretch of the imagination. That's going to require lots of energy to go through to input to reform that bond. Okay? So it is not something that we consider as reversible, whereas the denaturation is reversible because you're breaking and reforming relatively weak bonds, or weak interactions, I should say, weak forces, not bonds. As soon as we go through and break bonds, we're really pushing into that degradation process, not the denaturation. Okay? Questions on that? Okay. Chapter 5 is all about lab stuff. If you're interested in it, you're welcome to read it. I doubt any of you will really see much about it. Really what you could go through and do is apply your understanding of separation techniques in organic chemistry to biochem, and that's really all that chapter talks about. They just make all sorts of fancy new terms to confuse you. Okay, so if we try to isolate this compound, so let's think back to OChem. What kind of purification techniques did we have?
Extraction.
Okay, we could look at extraction.
Distillation. Chromatography.
Okay, so slow down. Extraction. I heard distillation. Column chromatography. What else? Crystallization. Anything else? Awful technique for purification, but we could do TLC. Okay. Not a great technique because it's very small amounts, but it does work. Okay, so we've got all these variety of techniques. How do these techniques all work? What do they separate based on? Forces. Okay? And extraction is arguably a little bit more than forces because that's actually looking at acid-base chemistry. Okay, but everything else is based off of forces. So how might we go through and take advantage of those forces to isolate individual proteins or amino acids away from each other? Are their forces different? What's that?
Polar forces.
We've got polar interactions within our amino acids. We have nonpolar amino acids. Okay, we have acidic amino acids. We have basic amino acids. Those have inherently different structures. What is that going to do for separation techniques? Well, if they're inherently different, can we separate them based off of their forces? Absolutely. The primary one that biochemists tend to rely on is this column chromatography, sitting in the middle there. We can potentially do extraction. Distillation is out because distillation requires the separation of liquids. Typically, we don't separate any solids with distillation. Okay? And with biochem, it's going to become even more problematic because you'd have to get it up into the gas phase to do that separation. Most biological things aren't particularly happy moving into the gas phase and usually are destroyed before they get there. Crystallization can work, and when you're looking at larger proteins, one of our techniques was X-ray crystallography. Okay, so we do need crystals, so we can go through and do that. Typically, to do X-ray crystallography, you do need a crystal. It needs to be a super pure crystal. So when you start making the crystal, it's already in a really, really pure state, okay, before you initiate that crystallization process. So we don't really use that as a purification technique. TLC also shows up, but TLC is really just a small-scale version of column chromatography. So column chromatography and TLC, are you guys aware of the basic principles behind those? Or should we talk about those?
We should talk about.
Hey, if you use a TLC plate to separate a compound like that, not only, once you have the separation, you're gonna, I don't know, cut out that little powder alumina. And you're gonna wanna basically use an extraction technique to separate your actual compound from the TLC.
Yeah.
So that's more work.
It's a lot more work. So what he's talking about is in column chromatography, because I don't think a picture is going to work like this. Column chromatography and TLC. So TLC, we effectively take a sheet of paper or a sheet of glass. It's usually a sheet of glass, and we coat it with a very polar material. Okay, so we've got a thin sheet or a thin layer of that polar material. So what happens? We spot our compounds on the bottom of it. We stick it into a solvent. The solvent moves up, and as it moves up, it pushes the compounds. Okay, well, the compounds interact with the polar stationary phase. Okay? The more polar the compound, the stronger the interaction with the stationary phase, which means what about its mobility? It moves less because it's interacting with the stationary phase. And the stationary phase, by definition, doesn't move. Okay? So TLC, you've got that very thin layer of silica over the top of it, or that polar material allowing you to try and separate. Okay? And that's just based off of polarity. How polar are your compounds? Well, we could evaluate and go back to all of our different amino acids. They all have the acid and the amine aspect to them, and they have that R group. Okay? So the acid and the amine really aren't going to be any different. The only thing that would be different would be the R group, and we could kind of separate based off of the R group polarity. But again, you're dealing with a very small system, okay? Only a thin layer on top of this. So what column chromatography does is it takes that TLC tube, or that TLC plate, and it turns it into a tube. What do we put through the middle of the tube?
You put cotton, you're gonna put your
I'm making it
solution.
more difficult than you need to. I don't need the exact details.
Non-polar.
Silica. You just fill it with more silica. So now instead of having a very thin layer of material to separate on, you have a lot more. The more silica you have, the more you can now separate. So you can put a larger sample size onto a column than you can onto a TLC plate. This runs into an issue though. If we have a very large column and we try and stick it into a solvent bath, the solvent's not going to run up very far. So what do we do instead? We pour the solvent down the top. Okay? So now instead of running from bottom to top, we run from top to bottom. Okay? So we dump our solvent in, and we just constantly run solvent through until everything comes off the column. Okay? The first compound off the column, should it be the most polar or the least polar? It should be the least polar. Remember, column chromatography is the reverse of TLC. We've just done it upside down. Our solvent runs downwards. So the least polar compounds can interact the least with our polar stationary phase, and it's just going to flow straight out of the column. Okay, so that's the basic principle behind column chromatography and silica. So we can do that with some amino acids and some proteins. Okay, but there's other things that we can go through and tweak. Okay, we can take advantage of that third pKa with a lot of our amino acids. And we could run it in a basic environment. Well, under a basic environment, what's that going to do to our acidic side chains? So we take an acidic side chain, take an acidic side chain right here. What happens if we run under a base environment? Gives up the hydrogen and it becomes charged. Wickedly polar. So if we now run our column with a basic solvent, anything that's remotely acidic is now stupidly polar and is now stuck to the column. Anything that is not as polar runs off the column. What if we ran under an acidic environment? Well, that puts the hydrogen back on. We're now looking at not much of a polarity difference for our acidic groups versus anything else. But what about our basic groups? The basic groups pick up that hydrogen and become charged. Once they're charged, they're now wickedly polar and they stick to the column. So we can change the polarity of our solvent by changing its acidic or basic characteristics to allow our compounds to either separate more quickly or slowly, depending on what things we're trying to separate. Okay. We can push this even further. You remember that whole R-A-N-T-Y, why not, anti, versus, say, G-N, shoot, I don't know all my letters very well. So now we have two short protein molecules. How could we take advantage of separating these? Now it's not just an amino acid. What could we go through and do? Pick a pH. What's the charge on this molecule at that pH? What's the charge on the other one? If one of them happens to be neutral and one of them happens to be charged, you now have a significant way to separate them, okay, based on those differences. How do you figure out all that charge stuff? That goes back to looking at the pKa's for all the side groups. You can look at PIs. You can look at all that fun jazz to try and determine ways to separate it. All right? So that's just really changing the solvent. We can also change the stationary phase. Okay? We don't have to use silica. We can use other solid substrates that have a higher affinity for one amino acid or another. So we could put in a hydrophobic stationary phase. Okay, so if it's hydrophobic, what's going to happen when a polar amino acid hits the top of it? It's going to move away from it as fast as possible. So now our polar groups move faster through the column because we've changed the interaction with our stationary phase. Okay, so a lot of what Chapter 5 is talking about is what are the different ways that we can take advantage of intermolecular forces to allow things to separate. It's all based off of intermolecular forces. We would do these techniques in lab, or at least some of them. You've probably already done some of them in biology labs if you've taken biology labs. Okay.
Isn't there a whole class on this at MCC?
MCC has a biotech class, and they do similar processes. So they go through and they'll pick individual sections and push through different purification schemes. Sometimes they work, sometimes they don't, but that's also the nature of biochem. Sometimes it works really well, and sometimes it doesn't. So now what we're going to talk about is, at least the tail end of Chapter 6 is probably my least favorite chapter because it confuses the hell out of me, and I just have a very hard time visualizing and organizing all the different terms that come out of this. Okay? And ultimately what this is going to come back to is kinetics. Okay? Enzymes are catalysts. So now that we've built up to proteins, we can look at what some proteins do. And this is where we have our enzymes. They are biological catalysts. So if we had, say, this reaction, what would an enzyme do to this energy diagram? What would it change?
The energy activation.
It's going to change the activation energy. How does it change the activation energy? Okay, it does lower it, but does that mean it just does this? Typically not. Okay, the enzyme lowers the activation barrier by changing the mechanism by which the reaction works, which means instead of having just one hump, it may have 2 or 3 or 4 or 5. But each of those steps will be a lower activation barrier than the overall one. So those steps, even though there's maybe more of them, occur faster than just going over the whole thing. Okay, so we have to drop that activation barrier. And the other aspect is that we really do change the mechanism. In that response, you'll notice that we do not change the energy of our reactants and products. Okay, which means everything that we talked about in chapter 1, as briefly as we did with the thermodynamics, remain unchanged. Which is why when we talked about this figure, the thermodynamics is just in this bottom half, and our kinetics is in the top half. Okay, because that's where those differences are kind of defined and separated. So in 152, if you took 152, you spend at least a chapter talking about kinetics and potentially different applications thereof and how you kind of piece it together. And a lot of people tend to not like that section and just kind of hope they can float through it and ignore it. It comes back. Right? Those of you that have not taken 152, none of this will look familiar. Right? I kind of apologize for that, but we have to do it. It's part of the material, so we've got to get through it. So I'm going to attempt to spin it in such a way that you can get just to the nuts and bolts of it and not have to stress too much about all the other things. Okay? So our primary difference is that activation energy. What's going on with temperature? So how else could we affect the rate of this reaction? Okay. Temperature seems to have an effect, but what does temperature ultimately do?
Speeds up.
It speeds up all reactions. Okay? And because what it ends up doing is providing more energy to both the reactants and the products so that their energies relative to each other stay the same, but relative to the activation barrier gets smaller. Okay, so they all shift upwards. Okay, that gives each of our particles a little bit more energy to clear the activation barrier, so we see the reaction happen faster. Okay, so it's not really a catalyst, but it does speed our reaction. Okay, much kind of like the idea of an enzyme, except the enzyme drops the activation barrier, temperature increases the energy of your reactants and products so that you can get over the activation barrier. Okay. I don't think I want anything else on that. Okay, there it goes. So we can look at kind of the qualitative discussion on this. So if we take a look at a standard enzyme, our enzyme will grab a substrate, because we need the substrate to interact with the enzyme so that the enzyme can then do whatever it needs to do. So we end up starting off with an enzyme and a substrate. Those two things interact with each other in some fashion. The enzyme facilitates whatever reaction needs to happen within the substrate and then spits out the product. So we've got two possible ways that we could look at this. You've got the method that's known as the lock-in key. And what's happening within the lock-in key, we look at the substrate and the active side of the enzyme, where the substrate needs to bind, you notice they look exactly the same. And that's the idea between a lock and a key. When you stick a key into a lock, it matches up exactly so that it opens. The other model is this induced fit. So what ends up happening is that the substrate comes in and binds, and then the enzyme changes its shape to accommodate the substrate. So if we look now at the enzyme's kind of pattern, you'll notice that all those edges now have changed oh so much and are now rounded versus the jagged result that they need to be to match the substrate. So that's kind of the idea between these two different mechanisms, either lock and key or induced fit. So lock and key, nothing really changes, it just binds, done. Induced fit, substrate comes in, enzyme sees it, changes its structure to account for the substrate's presence, and then we can move on. And then we've formed what's referred to as our enzyme substrate complex. Some other terms that are symbols, at least, that are going to show up. The substrate is commonly referred to as S. The enzyme is typically referred to as E. The enzyme with the substrate is commonly referred to as ES. And our product is typically referred to, although it's not shown here, as, what would our product be? P. So if we were going to write this out as kind of a reaction, we start with our enzyme plus our substrate, which goes to our enzyme substrate complex. After we form the complex, we can then go through to the product. So that's going to be kind of our overarching reaction symbolism that we'll go through. one of the things that we'll see get added into this is this extra arrow. The enzyme substrate can potentially decompose back into the enzyme and the substrate and not progress forward to the product. Okay.
Isn't that really how enzymes work though, is that they kind of, they are there at the beginning and they're there at the end?
Oh, so that
Right.
is in reference to actually me not finishing this reaction,
Right.
writing enzyme at the end. So this extra arrow isn't in reference to that. What this extra arrow is saying is that once the enzyme and the substrate bind and form the enzyme
They
substrate
may not
complex,
form the product.
they may not go forward to product. It may go backwards to just having the substrate in the enzyme. Okay, so that is a reversible process. Another aspect of this is that we also tend to, at least for biological catalysts, say that we don't have a reversible product or reaction once we form the product. Okay, that only forms the product. Once it forms the product, the enzyme does not bind the product and go backwards, okay, to go back to the substrate. This is a result primarily of our biological catalysts. Sometimes it happens with our natural or human-made catalysts, but not as often. Okay. So in the textbook's beautiful fashion, they're now going through and saying once we've got our enzyme substrate complex, we can form the product. You'll notice that we now have the product that's missing a little section out of it, just trying to represent a different structure, and it can then spit out that piece. So it's trying to represent this reaction scheme in kind of these pseudo-pictorial diagrams that in theory make it easier to understand. So we've got our enzyme plus our substrate to form our enzyme substrate complex. Okay, we've got the active site, which we already mentioned. And then the other thing that's going to become important, particularly when you talk about these things in your research project, is that our interactions are happening primarily between forces. Okay? There happens to be, say, maybe an OH here that matches an N there. Okay? Or maybe a valine here that matches a glycine there. Okay? So it's those similar interactions between those that allow those two things to come together. We're not forming a covalent bond here, it's just these weak interactions that kind of hold it. And as long as we have enough of those interactions, they can interact strongly enough to allow this type of thing to occur. Okay? Much the same way that if you've heard of chelation, anybody?
Yeah.
Okay, maybe let's try it the other way. Anybody not heard of chelation? Okay, wow, okay. Chelation therapy is this common thing that floats around in old folks' homes. Someone comes in with a bag of EDTA, which is a chelating compound. Okay, and all it does is it has 6 different sites on it that are all negatively charged. Okay, well, the idea behind chelation therapy is that there are toxic metals in your body. So we need to get rid of all of those toxic metals. Well, what charge do most metals carry? Positive. So if we come in and ingest some molecule that has a bunch of negatives on it, all of those negatives will interact with a positive. And if we have enough negatives interacting, we've now built a very strong interaction between the metal and your chelator. And in theory, we can now flush the chelating agent and the metal out of the body, and now you're cured of all your diseases because you don't have that toxic metal anymore. Problem with that theory is it's not selective for the metal. Okay? So while mercury may be very toxic, iron is kind of important. So if you ingest a lot of these chelating agents, it goes through and it very effectively grabs all metal ions, including the metal ions you wanted. Okay? And so it can become a toxic issue. So kind of an interesting process. Chelating agents are relying pretty much on the same thing that an enzyme is. Similar intermolecular forces between those systems, allowing it to properly connect and hold. Once it's held, we can actually do some chemistry. But it has to be able to hold. So we have to have some kind of pocket with proper intermolecular forces to allow for that connection. Because we also want that specificity. We don't want just every single molecule binding into that enzyme site. We only want the one that we're trying to get the reaction to occur with. So, interesting process here. PNP and DADME, don't ask me what those names mean because I don't know. Actually, PNP I know. Purine nucleoside phosphorylase. Okay, is a particular enzyme, and it metabolizes the destruction of adenosine into adenine. Okay, so we've got adenine on the right and adenosine on the left. Okay, so what the enzyme does is it grabs a hold of adenosine and facilitates breaking off of this bond. Okay? That bond it has to go through and break. And once it's done that, then you can spit out the adenosine and you can spit out the sugar. So if we're going to try and design a drug to target this particular enzyme, what should we be focusing on? Well, it depends how the enzyme grabs the molecule. So if we don't know, what we can do is do kind of a shotgun approach. We can take a look at this structure and say maybe it's focused on the sugar. So I'm going to provide a bunch of sugar molecules. Okay? Maybe it's focused on the adenine aspect of this. So I'm going to provide a bunch of molecules that look like adenine. Okay? But there has to be some variation off of it. Why do we need that variation? Well, if it's exactly like adenine, what did the enzyme do with the adenine? Spit it out. Well, if it's spit out, the enzyme's still functional. So what we end up doing is doing slight alterations to those individual structures. And what we're hoping is that when we go through and test these, that the enzyme activity decreases. Okay, so we continue to do slight refinements off of the structure to see how that affects the overall enzyme function. Okay, that looks like a link, but I don't remember where it goes. Should we go ahead and see it, hit it and see what happens? Oh, this is actually ATP synthase again. What they're trying to show is the enzyme side chains in yellow. Okay, and what we've got here is ADP kind of flipping and rotating in. Remember, everything is in motion. Nothing is static. Okay? And what will end up happening is once it's in its proper conformation, let's see. Last time I did this, I thought something floated in, but it may be that. Once it gets into its proper conformation, it can then go through and actually process the chemistry that it needs to do, which in theory it'll do here shortly. Do it. Not quite actual footage. And then slow, you're right. It is absolutely slow. Right? At least when we try and visualize it here. Realize that this is actually happening at a much faster rate in the cell. Okay? So we're actually seeing kind of a model of what's happening there. Everything is constantly moving. All of those interactions are occurring. That substrate had to adjust and shift its way through there, looking for individual interactions. You notice that once it went in, it didn't really fall out. Okay? And that's because it started to find favorable interactions within the enzyme to keep it locked in there. Once it finally hits its proper spot, then chemistry can start to occur. And now the structure's changed. Well, if the structure's changed, it's now going to change its affinity for that pocket and get kicked back out. Okay? So it's kind of a neat process. So we got another example for an enzyme. Chymotrypsin catalyzes the cleavage, let's say cleavage, I did go through and edit these slides, and for some reason I didn't catch that, of the carboxyl side of the phenyl and tyrosine residues in a peptide. Any guesses as to the characteristics of the enzyme? Any ideas? So what might we expect to see within that enzyme that would allow for this relatively selective cleavage? It's only cutting the carboxyl side of the phenylalanine and the tyrosine. So what characteristics of phenylalanine and tyrosine are similar? You all have those amino acids memorized, so you know what their structures look like?
What did it only cleave? Wait.
So what we've got, phenylalanine and tyrosine. What do they have in common that could potentially allow for some? The R group. We're looking at that ring. Okay? That ring is in common between those. Okay? It is quite likely that if we looked at the enzyme pocket for chymotrypsin, it probably has a relatively large size hole to fit that phenyl ring. And it potentially has even other rings that can then stack with that, okay, to facilitate and hold the amino acid sequence there. Once it's held there, it can now go through and cut the carboxyl end. Right? But it has to be able to have some affinity for those particular things. taking it a step further, which side of the enzyme? So if we assume that, okay, that's our enzyme, or sorry, our sequence, and this is our enzyme. Okay, that's behind it. Okay, which side of the enzyme should the pocket be on? Does it matter? Okay, that pocket is absolutely going to be critical. Okay, why is it going to be critical? We have chiral molecules in our proteins, which means when we grab the hold of that molecule, the phenyl is only on one side of the amino acid chain. So if this is going to be selective, it's going to make sure that that space is only on the one side where we would expect it to show up. Because if it's on the other side, if our amino acid went in backwards, it would cut the other end, the amino end, not the carboxyl end. Okay? So the direction in which or where our pockets are located is also of critical importance. Not just do I have a hydrophobic pocket, okay, but on what side of the enzyme is it located. Looking at those interactions is one of the things that we can go through and do when we're trying to target, whoops, go further back, when we're trying to build drugs. Okay? Someone may have already crystallized the protein, okay, that we're trying to target with whatever enzyme or whatever product is in it. Okay? So what we can then go through and do is look at the amino acids around that section and say, okay, how can I make a stronger interaction than the substrate or the product? And if I can make that stronger interaction, then I can target a molecule to bind in irreversibly. Okay, so it's looking at that structure and trying to determine something about how we can alter the structure and its interactions to get our desired result, which in this case, what we've been talking about is shutting down an enzyme. Okay. So chymotrypsin is particularly famous or infamous for actually looking at amino acid sequencing. So what we can do is take a big long amino acid chain, we can throw it into a soup of chymotrypsin and watch the chymotrypsin cut it up into smaller pieces. We can then analyze those smaller pieces. Okay? So a nice fun test question that you could potentially see in a more extended class, it would be something along these lines. What pieces would be isolated after the treatment with chymotrypsin? That's a fun question, isn't it? What does that require that you know? You need to know, and I would argue it doesn't mean you need to know all the R groups. It means that you need to know what the symbol is for phenylalanine and tyrosine, the one-letter codes. So where are those one-letter codes? Because you got them all memorized, right?
Y and Q.
Q. Okay. So what pieces would we isolate? Remember, where does the chymotrypsin cut? On the carboxyl end. In our amino acid, what do we start with? The amino end, and we end with the acid end, which means what we would end up with our pieces, which I didn't think this through carefully enough. It's not too far off, right? There's no U, so cut this here. Yeah. Okay. So what we're looking at is where does that chymotrypsin actually go through and cleave, and we can now see those individual pieces. And I tried to be fun and spell something, but I forgot. We don't remove it. It's still part of the chain. Okay? Questions on our chymotrypsin? It's not entertaining for me. I don't know what else to do. Okay, so kinetics review. Now is where we're actually getting into the belly of the beast. So given this reaction, A plus B goes to C plus D. Describe the rate of the reaction with respect to, so that's WRT, with respect to the concentration of A or the concentration of D. Right. So So those of you that have had 152 should be able to help out and chime in here. Not to call you out or anything, Jay or Victor. Or I think Teresa, you had 152 too, didn't you?
A year and a half ago.
It's okay.
You should remember it better than me. I had it five years ago. Well, it depends if it's first order, second order, or zero order.
It does not. So this is going to be one of the things that I think 152 people might actually have a disadvantage at because you're going to overthink this. And this initial question, it has nothing to do with first order, second order, third order, or any of that. It's not the rate law. What is it? Nope. You're starting to jump into equilibrium constant.
Rate is equal to K, C, D. It's your little concentration.
Nope. It's going to be the change in concentration of A with respect to the change in time. Right? That's it. What's going to happen to the concentration A with respect to time? Should it increase or decrease?
Decrease.
It decreases. Okay, so if I now also want to compare this to the concentration with respect to D, I can look at the rate as the change in concentration of D with respect to time. Well, what am I going to run into as a problem?
Aren't they inversely related?
They're inversely related. The change in concentration of D with respect to time is going to increase. That's going to be a positive value. A will be a negative value. If I want to rate, we want to avoid negatives as best as possible. The only way we can avoid negatives is bring in a negative. So what I can do to set all of these things equal to each other is that my rate will be negative of the rate of change of any of my reactants with respect to time. Okay? And that'll be equal to the change in concentration of any of my products over the change in time. Before anybody jumps me to it, if I go through and put in these extra symbols, that does cluster F this thing up. Okay, so let's ignore that for the moment, just not kind of stress on that aspect because it's not that big of a deal. Okay? So what we now have is a way to represent our rate. Okay? So now we're going to switch it out again and say what is the dependence of the rate with respect to just concentration. Okay? What's the dependence of the rate on the concentration of A versus concentration of B versus concentration of D? So now what I'm saying is not rate is related to the change. Okay? What is the rate exactly in comparison to A, B, and D? And theoretically, simultaneously. Okay? To do this, what we would have to go through and do is run an experiment. We could determine our rate by looking at those expressions that we just did. How does our concentration change with respect to time? And we can plot that versus our concentration. So in this case, I'm plotting technically just A, because that's all my axis says, but if we just said that was A, B, and D, okay, are all on this plot somehow. So let's go through and actually switch this. This is just our concentration of X. I'm going to go through and say that our blue is our concentration of D, our red is our concentration of B, and green, which takes me a second, and I also don't think it shows up nicely, there's our concentration of A. Oh, that showed up okay. So how does my rate depend on each of those concentrations? Any one of those curves easier than the other, you think?
D.
D. Why? Does the rate depend on the concentration of D? No. So if I'm going to look at my rate, okay, it has zero dependence on the concentration of D. So if I want to go through and do this, okay, how can I change my equation to now show that there is zero dependence on it? What I'm saying is that our rate will change for an individual reaction, okay? But it does not change with respect to D. So if I'm going to set up this kind of proportionality, if we set it up directly, if I change D, what happens to the rate? According to this proportionality, as written, if I change D, what happens to rate? This proportionality, not down there, right here. If D goes up, so let's say what rate is, okay? Tell me what rate is for a concentration of D. D is 1. What is rate? 1. What is, if D is 2, what is rate? D is 3, what is rate? Is there a relationship then? What happens to rate when I change D if this proportionality is true? It changes. Is that what we see? No. So we need to fix this expression. We need to add something in here that changes D in such a way that the rate will never change if D changes. What do I mean? What if I did plus 1? It will be a concentration of D plus 1. If I change D, does my answer change?
Yes.
Yeah, okay, so that doesn't work. Maybe I mean minus 1. Does that work? No. What can I do to this? It's not going to be a fraction. It's going to be a power, yes. What power is it going to be? 0. Because anything, okay, to the 0 power is 1. Does the rate ever change then with respect to D? No. That now matches my curve. I'm saying there's a 0 relationship there. Okay. What happens when we look at B? How does it increase? Linearly. So if I'm now going back to the same idea of rate is proportional to B, what is that power going to be? 1. Because it's going to be a 1-for-1 change. Okay? That's going to give me that straight line. Okay? And a straight line that will increase, not remain flat, not remain constant. What happens with A?
It's exponential.
Okay? It's exponential, and what we'll find is that the power is 2. How do we determine all of those numbers and those relationships? Okay, well, in theory now, we have programs that can go through and solve and fit the data and tell us what the equation is or what the relationship is. Okay, in the past, they didn't have that relationship, so they had to go through and try and convert everything into a linear format. And once we were in a linear plot, then we can interpret and say, okay, that's what this relationship is, that's all that fun jazz.
Can you just base the power on the slope?
It's not quite the slope, because if we base it just on the slope, what's the slope between 1 and 3, or 1 and 2 on the green line? Okay, relatively small. What's the slope between 10 and 11?
Consistent.
It's not consistent. So the slope only works if it's a straight line. If we get a curve, it doesn't work anymore. Okay? That's why we had to switch things around to get it into a straight line. And we can still do that. Okay? That is possible to do algebraically. So for this individual expression, what we can then go through and do is say that, well, D didn't matter. Okay? So all of these affect the rate, and all by multiples. So what I can do is simplify this down now into rate will be equal to concentration of A squared times the concentration of B to the 1 times the concentration of D to the 0. That's a 0. Okay. We're still going to run into a slight problem here. Let's see, did I do it right? Yes, I did. Okay, let's assume we don't change the concentration of A and we don't change the concentration of D. Our rate will be dependent solely on what? Just the concentration of B. Okay? So if this equation were true, if B is 5, what should my rate be? 5. So let's go back to my equation, my graph. B is 5. What is my value for my rate? 10. Interesting. It's not exactly the same. Okay? But it does vary by a constant. No, because the concentration of A was constant. We didn't change it. It varies by a constant. If it didn't vary by a constant, we would see this curve bend. It wouldn't fit a straight line. Okay? So it is varying by a constant. Well, what is that constant? I don't know. Let's make it up. Okay? We now have a rate constant. And what we have at this bottom section now is what's known as your rate law. Okay? D to the 0 is 1. What happens if I change the concentration of D? It's still 1. So would it make sense to write this expression instead as times 1 times 1 times 1 times 1? Should I go through and do it like that? No, why not?
Extra work, it's the same.
Extra work doesn't do anything. So what can I do with the concentration of D?
Take it out.
I can, whoops, that's supposed to be an eraser. I can completely remove it from my expression. One of the things that we typically find is most circumstances, products do not affect the rate of a reaction. Not true for biochem in a lot of situations, but in most circumstances, we don't see the rate affecting this, or our rate being affected by our products. So it typically has to do with our concentration of our reactants and to certain powers. So now the question is, what do those powers mean? So I think that's what's going on here. These are typically referred to as your reagent orders. Okay? So if we looked at an individual expression, the order of the species kind of refers to its importance. If it's 0 order, does it show up in the rate expression? No, because it becomes 1. So it is completely meaningless. It's a bystander to the reaction. I don't care about it. Okay? What if the order is 1? Okay, well, now it's involved in that reaction. Okay, it is part of that process. What if the order is 2? Okay, we're increasing its importance in the overall reaction. I need to have 2 of those molecules for this reaction to process. And 3, it shifts to now being it's all me. Why?
It runs the show.
Why does it run the show?
Because it's exponential upon exponential.
Think about the chemistry that has to happen here. These refer back to our reaction and saying how many are involved in our slowest step of our reaction. So if we have 1, okay, just 1, our rate law became just simplified down to that, where m was 1. Okay, well, that is still all A. Okay, and what we're saying by that rate law is that in our slowest step, the only thing that's important is the concentration of A. As we move further up in our orders, we become more and more involved in that. Why does 3 become it's all me? Because when it's 3, we're saying we need 3 molecules all to interact with each other, right? At the exact same instant. How likely is that?
Yeah, I don't think that's common.
That's not very common at all, right? And that's one of the reasons why we jump to it's all me. Because if we hit 3, we're saying that everything about that reaction is settling on that 1 molecule. And we have to get ultimately, as a rough approximation, 3 of those 1 molecules to interact with each other simultaneously to allow for the reaction to occur, okay, as an approximation. Okay? Well, the odds that we're getting 3 things together to do anything, okay, not very likely. The fun analogy that I go back to is relationships. You got single people, well, single people exist, that's fine. You got couples. You could get 2 people to come together and do things. That works out pretty well. How often do you get 3 things, 3 people coming together to have nice, harmonious relationships? Not very often. Okay? It just doesn't happen. Okay? Why? Because each of those things has differing interactions, and we have to match those up perfectly to allow it to occur. Okay? How do we determine those orders? Those of you from 152 might know this. You have to do an experiment. Okay? There is no other way to do it. You have to set up an experiment. So, if we go through and now take those basics and turn it into our enzyme kinetics, what we've got on this curve is our standard reaction. Substrate goes to product. Okay? What we're saying is the substrate is important in the overall reaction. We're looking at a first order reaction here. Okay? What might happen if we now add an enzyme? Why would it be steeper? Why, enzymes do what? Speed up the rate of the reaction. So even at low concentrations, we would expect our reaction to go significantly faster. Okay? But eventually what's going to happen? As we add more of our substrate, we've added now so much substrate that the enzyme can't interact with it. So what happens to the rate? It starts to flatten out. okay? And what we've now got is our standard kind of enzyme kinetics curve, okay? Speeds up the rate, and then eventually it flatlines out. Okay? And that last one, just because we've talked about it already, cooperative binding, what happens? As substrate binds, more binds, which means you increase the rate of the reaction. So we'll start out potentially going faster, but then it should do what? It should shoot upwards and then level. And we end up with that sigmoidal shape. Okay? So those are kind of your basics of your enzyme diagrams. What we'll be shifting into next week is looking at the hardcore Michaelis-Menten kinetics. Okay? It is not easy to go through and process, particularly when you then throw in the added layer of inhibition. Each of your enzymes can be inhibited and in different mechanisms. That will change how the enzyme functions. Okay? And what we just did with this curve, interpreting what we would expect out of it, you will have to do with inhibitors as well. Okay? So read up on it. You may review the slides if you want. Maybe those will help you kind of understand it. It's not going to be easy, okay? But that's where we'll pick up on Thursday.